Hello, everyone. Very good evening to you all. Good evening, everyone. How are you all doing today? So good to see people already joined here and waiting for the session. So I really hope and all our biotechnical team, we all hope that is going to help you all for preparing for the CSIR exams. So we are tackling the tricky questions from different subunits of Unit 8, that is genetics, okay, or the inheritance biology as your syllabus says. So can we have some quick hellos here? Good evening, Jasmine. Good evening, Tira. How are you people doing? You are most welcome, Sapna. As you know, Biotechnica, we always want to help the students. That is our foremost priority. Okay. So are you finding the classes helpful? Okay, I'm also doing good. Thank you. All right. So I'm very excited to see the questions, to explain the questions, and so that we can solve these questions from linkage maps today. Okay, I'm glad, Jasmine, that you are also finding it helpful. Good, good. Okay. So today's topic will be linkage map. Now, I really expect that all of you know the basics of linkage mapping, right? So linkage map is a way of creating the genetic maps, right? Now, genetic maps, so before we go in the detail, let me give you a brief idea about what is genetic map. So just like we have a geographical map, right? Google Maps सब लोग use करते हैं. तो गूगल मैप में क्या होता है? It's basically the different landmarks of a city or a town, and there will be ways shown how you can navigate from one place to another. Correct? So in the same way, जब हम जीनोम को देखते हैं, जीनोम बहुत वास्ट है, उसमें बहुत सारे जीन्स है, बहुत सारे जेनेटिक एलिमेंट्स है, right? So वो सारे genetic elements, where they are present in the genome, how we can move from one genetic element to another genetic element, okay? To put it properly at their particular locations is what is called as gene mapping, okay? Now this gene mapping can be done by two ways. There's genetic maps, which we do it by considering the genetic recombination frequencies, okay, between the genes, from between the genetic elements. And the other way is by physical mapping, where we actually find out the locations of the genes, okay? So like to understand it better, I can give you an example from our daily life, okay? So अगर landmark है, okay? So let's say there is a shopping mall near your house, okay? So अगर आप उसको किलोमीटर में बताएंगे, that will be a physical mapping, okay? Because distance तो किलोमीटर में नापी जाती है. But अगर आप उसको it will just take 5 minutes from my house to the shopping mall, okay? So that is like, time is not a unit of distance, okay? But here we are still using it to get an idea about the distance between two places, right? So that is what is a genetic map. Okay, I'm getting the input that the sound is not clear. I have full this. So, hello everyone. Is it any change, any better? Okay. All right. So yes, so what I was telling you, that when you explain the distance between two places in time, okay, which is not actually an unit of distance, okay, so that is like genetic map, where we create the linkage maps, okay, with the help of recombination frequency. Okay, recombination frequency is used to find out the distance between two genes, and when we use that as a parameter to map the genetic element on the chromosome in the genome, that is what is called as linkage maps, right? So these linkage maps are used for gene mapping, definitely, but when you see the CSIR exam questions, linkage maps are extremely important from exam point of view. So if you take last 10-year question papers, you will not find, I guarantee you, you will not find a single question paper without any question from this segment. It might be a part C. Usually it comes in part C, but there might be question in part B as well. So definitely you are going to find a question from this linkage map, from this entire gene mapping subunit. Okay, so that's why taking the entire gene mapping subunit together, I have further broken it down. So today we will have linkage map related questions, and next one we will talk about the molecular marker based questions, okay, which is a way of physical mapping, right? So let's get started here. So first question, so two genes A and B are located at a distance of 10 centimorgan. Individuals of genotype capital A, small a, capital B, small b are submitted. The two genes are linked in trans. What percentage of the progeny is expected to have the genotype small a, small a, small b, small b? Okay. So here also, I think in the previous class also I explained you what is submated. So submated means mated with an individual of the same genotype. Okay. Now the genotype is given here, but when we are talking about the genes which are linked, so if the distance between the gene is 10 centimorgan, means genes are linked. And if the genes are linked, then it becomes important that we find out which are the alleles of the genes that are present together on the same chromosome. So now it is mentioned that the two genes are linked in trans. Now trans and cis, they are the two ways in which the linked genes can exist. So trans means where dominant allele of one gene will be present with a recessive allele of another gene and on one chromosome, and in the other chromosome it will be the recessive allele of the first gene is present with the dominant allele of the other gene. Okay, so this kind of arrangement is called trans. Okay, whereas cis will be where dominant of both are present on one chromosome and recessive of both are present on another chromosome. Okay, so whether I write like this, okay, like how I have written, like numerator denominator, but it's not a division, it's just showing that these two alleles are present on one chromosome, these two alleles are present on another chromosome. Other way of writing it will be like this, separate with a slash. Okay, so in either of the way you can write the linked genes. Now let's see the cross here. So cross is this. Okay, now what do we want? We want a genotype of small a, small a, small b, small b. Okay, so how do we create it? So small a, small a, small b, small b, if we have to write in this linked gene format, it will be small a, small b, one chromosome, small a, small b. Okay, so how can you get it like this? When you get a small a, small b chromosome from this parent, small a, small b chromosome from this parent, and they fuse together, then only you can get an individual of this genotype. Okay. From here, only when there is crossing over occurring here. Right? Now, what is this 10 centimorgan? 10 centimorgan is talking about the distance between the genes. Now, this distance between the gene is about the recombination frequency, right? So recombination frequency percentage is what is equal to 1 recombination frequency percentage. 1 recombination frequency percentage is equal to 1 centimorgan, right? So if there is 10 centimorgan distance between the genes, between That means recombination frequency between these two genes is 10%. Okay, but 10% will include both the recombinants. Now, when there is recombination here, so what all recombinants we will get? So this is the recombinants. Okay, so recombinant can be capital A, capital B, and small a, small b. Okay, total their recombination frequency is 10%. All right. So what will be the proportion of small b only? It will be half of 10%. Namaste, Amit. You are a little late, but still we are discussing the first question only. Welcome to the session. Okay. So what will be the frequency of small b? It will be half of 10%, that is 5%. Okay. So how much will be, what will be the proportion of, or what will be the chance of getting small a, small b? It will be 5%, or in terms of decimal, it will be 0.05. Okay. Similarly, what will be the probability, or what will be the proportion of small a, small b from this parent? In the same way, it will be 0.05. Okay. Now, when we are talking about this individual, so what will be the probability of that? That will be equal to, remember the multiplication rule, where we have to multiply, because both of them have to come together individually. They both are independent events. They have to come together, and then only we can get a genotype like this. Okay. So it will be probability of getting small a, small b from one parent multiplied with probability of getting small a, small b from another parent. So here this is 0.05, and here this is 0.05. Okay, so it will become 0.0025, which in terms of percentage. Okay, why? Because in the question it is asking percentage. So in terms of percentage, it will become 0.25%. So answer will be option 1. Okay, so like this, depending on whatever distance is given here, we can do the calculation. All right, so just here you have to find out whether the genotype that it is asked, whether it will be created by the recombinant or by the non-recombinant. Now moving on to the next question. Now, this is also a similar question, similar type, okay? But again, you have to understand the concept here. So it's given that in a linkage map, two genes, capital A, capital B, are 70 centimorgan apart. If individuals heterozygous for both the genes are test crossed, number of progeny with parental phenotype will be, options given, equal to the number of recombinant phenotype, more than the number of recombinant phenotype, less than the number of recombinant phenotype, or could be more or less than the number of the recombinant phenotype, depending on whether the genes are linked in trans or cis respectively. Now, to understand this here, okay, so you have to remember the concept that in linkage map, what we are doing, we are finding the recombination frequency. Now, recombination frequency between linked genes, the maximum recombination frequency between linked genes that we can calculate is 50%. Okay. Now, once it becomes 50%, then the genes are becoming independent. Okay. So the genes that we see the Mendelian crosses, right? So in Mendelian cross also we get 50% recombinants, 50% non-recombinant, the parent, right? And that is independent assortment, right? So the maximum recombination frequency that we can calculate by this linkage map is 50%. Okay, so anything, so as soon as it reaches 50%, then we cannot associate with the linkage. So we cannot tell exactly whether the genes are present on the same chromosome or they are present in different chromosomes. Why? Because once the recombination frequency becomes 50%, that means the genes are independently assorting. Okay. Now, if the genes are independently assorting, okay, so even genes present on the same chromosome are also can be independently assorting, with the idea that if the genes are basically present at very far distance, okay, to opposite tips of the chromosome, then also they will be independently assorting even though they are present on the same chromosome. Okay, so basically independently assorting means what? That anytime that particular chromosome is entering, crossing over, at least one event of crossing over for sure will happen between those two genes. Okay, so it's not that independently assorting genes has to be present in different chromosomes. Even two genes present on the same chromosome can also be independently assorting if they are very far apart. Okay, so till the genes are linked, that is when the recombinance frequency will be lesser as compared to the parental's frequency. Okay, but when we see that the genes are 70 centimorgan apart, means 70% is the recombination frequency, which is greater than 50%. Okay, what does it tell you? That A and B gene, they will not behave as linked gene. Instead, they will behave as independently assorted genes. Now, if the genes are independently assorting, then if the individuals are mated, so that means Mendelian inheritance pattern, right? So in Mendelian inheritance pattern, if you take a heterozygote and do a test cross, what kind of progeny do we expect? Parental is recombinant will be equal to 1:1. Yes or no? In Mendelian test cross, what do we expect? 1:1 ratio, right? What is 1:1? Parental is to recombinant. Okay, so if the genes are 70 centimorgan apart, so basically anything about above 50 centimorgan, also if it is given, then also the genes will assort independently. So in that situation as well, in this test cross, the parental phenotype, the progeny with parental phenotype will be equal to number of progeny with recombinant phenotype. Answer will be option 1. Did you all understand? Come on, guys. I'm not getting responses from you all. Okay. So again, as you know, as the pattern of the sessions go, so write down your query, and then at the end of the session, you can join the Zoom class and get your doubts cleared. Okay. And stay till the end, and you will get some freebies as well that will definitely help you in preparing for your exam. Okay, and also at the end we will have the Zoom class as we are always having, and we can do the one-on-one doubt solving. So whatever queries, whatever doubts you are having, please note it down so that you don't forget. Okay, yes, next, what do we have here? Next question. So E. coli cells are simultaneously infected with two strains of phage lambda. One strain of phage has lambda phage had a mutant host range, is temperature sensitive, known to produce clear plaques. Genotype is HSTC. Another strain of lambda phage carried the wild type alleles, genotype H plus ST plus C plus. Phenotype, progeny phages were collected from the lice cells and were plated on bacteria. The following numbers of different progenies are obtained. Okay, so we have the progeny phage genotype and we have the number of plaques given. Okay, what will be the order of 3 genes and the map distance between them? So this is a typical linkage map related question of 3-point test cross that is asked in the exams. Okay. Now let's see this question here. Okay. So basically what we have, that inside the bacterial cell, these two lambda phages will come with each other and they will undergo recombination. Okay. So what is the genotype that we have? So on one chromosome, we have H plus, ST plus, C plus. Other chromosome, we have H, ST, and C. Right? Okay. So these progeny phages are collected. So the recombinant progeny phages are collected and were plated on bacteria, and the following number of progenies we have got. Okay. In this scenario, so when you get a table like this, so first thing, what do we do? So we have to find the order of the three genes. Okay. So for order of the three genes, it's important us to find the middle locus, because as soon as we find the middle locus or gene present in the middle position, then we can have the order. Okay. Now for that, we have to go a step-by-step manner. So first thing that we do is we find the double crossovers. Okay. See, what are double crossovers? So basically, if you have, just draw it here. So if you have three genes, okay, H, ST, no, I'm not writing with H and ST. So let's write it with three genes like A, B, C. A, B, C. Okay. Other chromosome, you have small a, small b, and small c. Right. Now, double crossover means what? That one crossing over event is happening between A and B, and another crossing over event is happening between B and C. Okay. So that will be a double crossover. Now, why do we choose the double crossover? Because what will be the result of the first double cross of first crossover? Okay, I'm writing it in red because I marked it in red. Okay, so only this crossover it happens. So the result will be A, small b, small c, one chromosome, and then small a, capital B, and capital C will come on another chromosome. Right? That will be result of the first crossover. Now again, from here, if another crossing over happens between B and C, okay, so this is after first crossing over, now after second crossing over. So what will be the result? Now you will get capital A, small b, capital C, and small a, capital B, small c. Okay, now when you compare this one with this, okay, so you see the genes which are present at the corners are same, capital A, capital C. What is changed here? The middle one. Here it is capital B in the parental combination, whereas in the double crossover, it has become the small b. Okay. Similarly, for the other chromosome, the chromosomes, the genes on the sides are same, small a, small c, but in middle it has got changed. Okay, so that's why if we want to find the order, we have to locate the middle gene. And for locating the middle gene, how are we getting it? By comparing the double crossover with the parental. Okay, so from that table, our first objective will be to find the double crossover. Now, in double crossing over, what is happening? Two events of crossing over has to happen together. Okay, and each crossover is independent of the. So because of that, the probability of double crossover will be lesser than a single crossover. Okay, so in fact, the double crossover products will be least in the table. Okay, so what will be the double crossovers? The least values. So what are the least values? This one and this one, 4 and 6, right? Means these are the double crossover products. Okay, so we have the parental variety written here. So this is parental. So on parental, one chromosome has all plus plus plus, other chromosome has the recessive alleles. Okay, now you write down the double crossovers. So double crossovers, what do we have? H plus, C plus, ST, and HCST plus. Okay, so now when you compare the double crossovers with the parental, what do we see? ST is the gene whose combination is changed here. Right? So if that happens, what does that mean? That ST is our middle gene. So we have found our middle gene, which is ST. Okay. Now, the funny part of this question, that this question is asked in part C. Now, definitely you feel that yes, it has to be in part C because both the things we have asked to find out, we have to find the order as well as the map distance. So it will be a lengthy process. So we have to, like, it should be in part C. Okay. But do you see the option here? There is only one option with ST as the middle gene locus. So this you can, if you understand the concept of 3-point test cross, so you can find it out in, I will say, less than a second, less than a minute. Okay, or probably in 1 minute only. So in 1 minute, if you are able to find it, so you see there is only one option with ST as the middle gene locus. So you don't even have to check for map distance. Directly you can go for third option, and that will be a correct answer. Okay. But yes, I will show you how to calculate the distance between the genes as well. So for calculating the distance between the genes, so we have to consider the single crossovers. Okay, so these ones that I have marked, they are the double crossovers, the least values. Similarly, from the table, the highest values. Okay, what are they? They will be the parentals, because linked genes, they will always try to remain together. Okay, so if these are the parentals, then what are the other ones, the other 4 categories? So these other 4 categories are the single crossover products. Okay, so now when we are calculating the distance between the genes, the first thing that we have to do is we have to rewrite the genes in the correct format, means keeping ST as at the middle. Okay, so if we keep ST at the middle here, okay, so it will become H plus, ST plus, C plus, and H, ST, and C. Okay, now let's find out the distance between H and ST. Okay, so for that we have to find out what are the crossovers that are created. Okay, so the single crossover products will be what? So if you create a crossover here, so H plus ST, C will be one crossover product. Other crossover product will be H, ST plus, C plus. Okay, now when we see the table, so when you check the number of strains with that particular genotype, so you'll find that 86 are for this and 114 for the other genotype. Okay, now when we are calculating the distance, so what we have to do, we have to add all the recombinants that have been created by crossing over occurring between H and ST. So definitely it will include the single crossovers, but at the same time it will also include the double crossovers. Why? Because in double crossovers, one event of crossing over is happening between H and ST. Okay, total number of cells was 1000. Okay, this whole multiplied by 100, that only will give you the recombination frequency percentage, and that will be the distance. Okay, so when you solve it, you will get as 21% as the distance between H and ST. Okay. Similarly, let's find out the distance between ST and C. Okay, so again, I will write down the cross so that we find out what are the single crossover products here. So H plus, ST plus, H, ST, and C. Now, second crossing over, if it happens here, so in that scenario, what do we have? The crossover products, it will be H plus, ST plus, and C. Okay, and the other variety will be H, ST, and C plus. Now, what are the numbers given from the table? Again, you can check from the table. So this is 90 and this is 50. Okay. Now, exactly just how we did in the previous case. So distance between ST and C will be equal to the single crossover products. Okay, 90 plus 50. But again, the double crossovers also need to be added, because one event of crossing over in the double crossover also happened between ST and C. Okay, so again divided by 1000 whole multiplied by 100 here. Right, so here you will get the combination frequency or the distance as 15 centimorgan. Okay, so distance will be centimorgan. Okay, so ST is the middle gene, and then the distance between H and ST is 21 centimorgan. ST and C is 15 centimorgan. Okay, so just for the sake of doing it, we did it. But for exam point of view, you could have solved this question in less than a minute, just by finding the gene for the middle locus. Okay, so that will be the smart way of solving. Okay, so always take the hint from the options. Okay, and that is one of the benefit of this multiple choice question, that you can take the hint from the options given. Okay, now next we have another question, and this is a very interesting question. Although it looks little complicated, but when we solve it, you will see that this is very interesting and very quite easy to solve as well. Okay, but conceptually it has to be strong. You have to be strong. Okay, and in fact, this question does not involve a single line of calculation. You don't have to do any calculation. Only thing you have to do is concept and analyze and link it with the question here. Okay, let's see here then. In the following pedigree, 3 STR loci A, B, and C are linked on the long arm of X chromosome in the order A, B, centromere, A, B, C, telomere. Further in the table, STR alleles present in each individual is indicated. Okay, so here is the table that is given. Now the pedigree chart is given. Okay, so you do not have to have a lot of knowledge of pedigree for solving the question. Some basic knowledge you need to have. First of all, circle means female, square means male. Okay, and I think that's it. Okay, so that is the basic knowledge that you need to have, and then we have to analyze it. Now this STR loci that we are talking about, okay, it is present on the X chromosome. It is mentioned that it is present on the X chromosome. Okay, so another thing that you have to remember, that males will have only one X chromosome, whereas females will have two X chromosomes. Okay, in fact, a hint is also provided to you that X chromosome in males will help you identify the phase of the alleles. Okay, that means how the alleles are together. So let's see how it will help. So based on the above X chromosome, in which of the following individuals are recombinant? Okay, that is what you have to find out. Right? Okay, now let's see. So first of all, do not get confused with these numbers here. So these numbers are nothing but the alleles. Okay, so if you analyze properly, so this is just like, we usually do not use numbers as alleles, right? So we use capital letter, lowercase letter with a superscript of plus. So that becomes easy for us to understand, but when there is numbers, we get confused. So do not get confused. Here the numbers are just the alleles. So basically it means that this A loci has 4 alleles. What are they? 4, 5, and 6. B loci has again 3 alleles, 7, 8, and 9. C loci has again 3 alleles, 1, 2, and 3. Does it make sense now? So 4, 5, 6 are the alleles of A loci. 7, 8, 9 are the alleles of this B locus, and 1, 2, 3 are the alleles of C locus. Okay, but they are alleles. They will occupy the same position of A, B, and C respectively only. It cannot occupy any other position. Okay, now let's write down the cross. Right. Okay, so from the so you have to find out who all are recombinants. Okay, so for that, let's find the genotype of this generation 2. Okay, now this generation 2, first of all, this second individual, who is he? Is a male. Okay, male have only one X chromosome, and these 3 loci are only on the X chromosome. So that's why, see, the males have only one allele. Okay, so if the generation 2's genotype, if I have to write in terms of linked genes, so how it will be? So this is the males that I'm writing. Okay, so it will be 6, 8, and 1. Okay. Other chromosome is Y. Why? Because this is a male. Okay. Now, what about the female here? This is a female. Okay. Now, female for them, for all the three loci, because she has two different chromosomes. Okay. Now, which alleles are present in which chromosome? So for that, we have to check her father. So who is her father? Her father is generation 1, 2 individual. Okay. So 1, 2. So he is the father here. Now, why do we have to check the father? Because father has only one X chromosome and he will give his X chromosome directly to his daughters without any recombination because he has only one X chromosome. Okay. So whatever alleles are present, 492, that means one X chromosome for sure will have 492. This is the chromosome that she is getting from her father. Okay. Now, if you take out 492 from this individual here, so 4 eliminate, 9 eliminate, 2 eliminate. Okay. So what are we left with? We are left with 6, 7, and 1. That means on the other chromosome, it is 6, 7, and 1 that is present. So 6 allele for A, 7 allele for B, and 1 allele for C. Now we have wrote down the genotype in linked gene format for generation seconds, 1 individual and 2 individual. Okay. Now let's see the third generation. So in the third generation again, okay, so we'll see each of them one by one. So in generation 3. Okay. So for generation 1 also, for the males, for sure, you can write down the genotype in the linked gene format. But for females, we cannot write it down because for writing it down, again, we need the information from their parents. Okay. Then only we can find out the linked genes, how the alleles are present in the chromosomes for the females in generation 1. Okay. So we're not going in generation 1, we are going in generation 3. So in generation 3, what we have? So we'll see each individual one by one. So for first, the first is a female. So she will have 2 X chromosomes. Okay. 1 X chromosome she is getting from her father. Who is her father? This male here is her father. Okay. So definitely that means 1 chromosome has 681 because directly from father she will get. So here, okay. Now one more thing here. See, only 1 X6 is given. Okay, so if only 1 times 6 is written, that does not mean that for A, only 1 allele is present. It means that both the alleles for A is 6. Okay, that's why only 1 times 6 is written. Okay, it's a female. Female will for sure have 2 X chromosomes. Okay, and both the X chromosomes will have one location for locating this STRA. Okay, so if only 1 times 6 is written, means both of them has 6 allele. So 6, we have written it 1 time, 6, 8, 1. So another 6 is left. So here it will be 6, 9, 2. 6, 8, 1 directly coming from father. Now 6, 9, 2 means for sure is coming from mother. Okay. Now in mother, does she have 6, 9, 2? No. She can form 692 if crossing over is happening here. Then only this 692 can be formed. Right? What does that mean? That this generation first individual is receiving a recombinant chromosome from the mother, recombinant X chromosome from the mother, which means that she is recombinant. Similarly, second. So once you have understood it, you can add. You can see it in all the 4 individuals one by one. So I suggest that you do it along with me. So second individual is a male. Male has only 1 X chromosome. So for male, 1 will be Y. The other is 492. This also the male is getting from mother. Does mother have this 492? Yes, mother has that 492. Okay, 492 is there in the mother. That means it is you getting the parental chromosome from the mother, means he is non-recombinant. For third individual, okay, it is again male. So he has only one X chromosome, which is 672. Now, does mother have 672 here? She does not. So again, how can 672 can form? If there is crossing over occurring between the B and C loci. So it will be, then it will be a 672. So 672 is getting, means he is getting from mother a recombinant X chromosome. Okay. Fourth individual is again a female. Okay. So for one chromosome, we'll straight away write down the genotype of the father for X chromosome. So it is 681. Eliminate 681. What are we left with? We are left with 6, 7, and 1. Okay. 6, 7, and 1. Does mother have it? Yes, mother has that 6, 7, 1. That means she is a non-recombinant. She is getting the parental chromosome only from the mother. Okay. Now the question is, who are all the recombinants? So recombinants are generation 2, 1, and sorry, generation 3's 1 and 3. Okay. So as I told you, there is not a single calculation you have to do in this question. Only thing you have to do is analysis. So answer will be this option 3. Again, guys, let me remind you that please write down your doubts so that you can ask me in the Zoom class session. All right. Now let's see one more question before we go to Zoom class and we can do the one-on-one doubt clearing session, as well as if time permits, we can see some extra questions as well. So I request you all to join the Zoom club, Zoom class after this question. Okay. Let's see one more question here. A 3-point test cross was carried out in Drosophila melanogaster involving 3 adjacent genes, X, Y, and Z, arranged in the same order. So the order is given to you. Not only the order, the distance between the genes are also given to you. X and Y is 32.5 map units, and that between Y and Z is 20.5 map units. Now the coefficient of coincidence is also given, which is equal to 0.886. What is the percentage of double recombinance in the progeny obtained from the test cross? So what all information are given to us? So the gene map is already given. So I'll draw down the gene map. So there are 3 genes located on it, X, Y, and Z. Distance between X and Y is 32.5 map units. And distance between Y and Z is 20.5 map units. Okay. Now there is something given as coefficient of coincidence. Okay. See, what is coefficient of coincidence? See, even though when we are expecting double crossovers to happen, so we are expecting that the double crossovers are independent of each other. Okay. But actually what will happen is they will not be independent and there will be some kind of interference. Okay. So one crossover will always interfere with the other crossover, the second crossover to occur between the same DNA molecule. Okay. So because of interference, whatever is the expected frequency of double crossover, we will not see. We will see a reduced number. Okay. Now, that reduced number is what is called as the coefficient of coincidence. So, coefficient of coincidence is basically whatever is the number of the double crossover, what percentage of the double crossover or of the expected double crossover that we are actually observing. That is called as coefficient of coincidence. Again, I will tell you, coefficient of coincidence is the measurement of what percentage of expected double crossovers we are actually observing. Okay. So if we see the formula for calculating coefficient of coincidence, so it is given by coefficient, okay, I'll just change the color. So coefficient of coincidence, which I'm writing as COC, is given by frequency of observed double crossovers divided by frequency of expected double crossovers. Okay, that will give you the value of coefficient of coincidence. Okay, now in this situation, coefficient of coincidence is given. What you have to calculate? You have to calculate this number, this value. What is the frequency or what is the percentage of double recombinants in the progeny? Okay, so you have to calculate this. So for that, this value is already given. Now, this value we have to calculate. Okay, how can we calculate that? So that we can calculate the multiplication of their individual crossing over frequency. So what we basically mean here is, so you have this gene, right? So you have the gene map. This is X, this is Y, this is Z. Okay, distance here is 32.5 centimorgan or map units. Here it is 20.5 centimorgan or map units. Okay, now what will be the frequency, or you can also write it as probability of expected double crossovers? Okay, so expected double crossovers will, so what is our expectation? That both the crossing over events are independent. Okay, so it will be probability or frequency of first crossing over multiplied with the probability or frequency of second crossing over. Okay, now what is this recombination percent? This distance is actually recombination frequency percentage. Okay, so if you take out, if you remove the percentage, so that will give us the recombination frequency. Correct? So what is the recombination frequency at the between X and Y? That is like remove the percentage. Okay, so it will become 0.325. Similarly, what is the recombination frequency between Y and Z? Again, remove the percentage, it will become 0.205. Now, when we multiply it, okay, so that will give us the expected frequency of double crossover. Okay, now you substitute that in the coincidence coefficient of coincidence formula. So which was frequency of observed DCOs divided by the frequency of expected DCOs. Okay, so expected, so here we have to calculate this value. So keep it on one side. So frequency of observed double crossover products will be how much? Coefficient of coincidence multiplied by this value. So what is this value? It is coming from here. Okay, frequency of expected double crossovers. So coefficient of coincidence is given to us, which is 0.886. Expected double crossovers frequency is 0.325 multiplied by 0.205. Okay, when you calculate it, you will get it as 0.059, which is almost equal to 6%. Okay, so how much will be the coefficient of, how much will be the percentage of observed double crossovers? It will be almost 6%. Okay, so whenever we are doing this linkage maps, it's very important for us that we understand the concept here. How do we calculate the distance? How do we find out the coefficient of coincidence? How do we calculate interference? Okay, so interference is 1 minus coefficient of coincidence. So if in any question it is asking you to calculate interference, so it will be 1 minus coefficient of coincidence. Okay, or in any question, if interference value is given and from there you have to calculate coefficient of coincidence. So coefficient of coincidence will be 1 minus interference. Okay, that's simple. So all these correlations, you should be clear with it. And not only these formulas, you should be clear with the concept behind it. Okay, because anyway the calculation is asked, whether the direct way like this or a back calculation is needed, you will be able to do it only when you are clear with the concept of this linkage mapping. All right, so with this I am going to end the live session here. I request the team to please post the link for the freebies as well as the link for the Zoom class. So everyone, I hope you have noted down your doubts. So please come in the Zoom class. We can have a one-on-one discussion. At the same time, we can also discuss some extra questions as well from linkage mapping. Because linkage mapping is such popular topic for CSIR exam that there is a lot of questions. Okay, definitely we cannot discuss all. We cannot even discuss 10% of those questions from the previous 10-year question paper. But I'm trying to do as much as possible. ठीक है, तो आप सब लोग जूम क्लास में आइए, वहां पे हम वन ऑन वन क्वेश्चन सॉल्व भी करेंगे और डाउट क्लियर भी करेंगे. All right, so I'm going to stop the live session here. I hope to see you all in the Zoom class to continue our discussion. Thank you, everyone.